Okay, so as I mentioned before, the title of my paper is Loss of Nicastrin from Oligodendrocytes Results in Hypomyelination and Schizophrenia with Compulsive Behavior. So, some background information that you're going to need to understand the paper. First, you'll need to know about schizophrenia. Schizophrenia is a very severe mental illness that's marked by positive and negative psychotic symptoms. So you can see some of the symptoms listed here: hallucinations, delusions, some negative symptoms, flat affect, reduced social interaction, etc. And then another thing that you'll need to understand is myelin and myelination. So, in this case, this paper refers to myelination in the central nervous system, which is carried out by cells called oligodendrocytes. And the oligodendrocytes, as you can see here, wrap myelin around axons, and this sort of speeds up the rate of transmission of electrical messages as they travel through your brain. So it speeds everything up in your brain, basically, and it's very important. So one of the issues that the paper talks about is hypomyelination. So that would be less myelin in your central nervous system, which can create a lot of issues, as we will see shortly. This one central theme of this paper is looking at what is the connection between these two things, schizophrenia and proper myelination. So one way to answer this and to look at this is by looking at the proteolytic complex known as gamma secretase. Gamma secretase is an enzyme like we've been learning about recently in class, and it has many different functions. One of the more well-known functions is to cleave proteins that are involved in creating amyloid plaques that are associated with Alzheimer's, but another important function of them is it has to do with proper myelination in the central nervous system and making sure oligodendrocytes myelinate axons properly. And it does this through interacting with certain substrates. And how these substrates that maintain healthy myelination work is they bind with the binding site on gamma secretase, which is known as nicastrin. So if you get rid of nicastrin, which is the binding site for these substrates, you end up getting hypomyelination because it can't interact properly with its substrates without its binding site. Okay, so here's going back into the connection. Is basically one of the common marker that is found in brains of schizophrenics is there is decreased white matter, which means there's less myelination, and they suspect that this may affect dopaminergic pathways and sort of mess them up. And dopamine is very important in schizophrenia because high levels of dopamine in some parts of the brain and low amounts of dopamine in other areas of the brain are thought to actually create the symptoms of the disease. So if the issues with white matter and issues with myelination is what is throwing this off, then that would be important to know, because that's going to affect what kind of treatment you're going to want to find. So a normal pathway for myelination in someone who does not have schizophrenia, you have the ligand neuregulin, which binds to its receptor ERB, and then this ends up triggering development of oligodendrocytes. So the oligodendrocytes, and basically tells oligodendrocytes to start, myelinating everything as they should. Now what happens in this altered pathway when you don't have nicastrin, so this happens in maybe like a schizophrenic brain, you are not having that substrate binding to the nicastrin, and so the process isn't being activated properly. So as a result of this, the ERB signaling is lost, and then because of this, the neuregulin remains active. It's not binding to its receptor properly, and then as a result of this, you end up not getting correct development of the oligodendrocytes. The oligodendrocytes don't myelinate the way that they should be, and you end up getting hypomyelination and loss of white matter. So what this specific experiment looks at is if the gene that codes for nicastrin is knocked out in mice, will they develop schizophrenia-like symptoms and myelination patterns? So This is to really confirm that nicastrin and this signaling pathway and gamma secretase are very important when it comes to having proper myelination and not developing psychiatric disorders like schizophrenia. And then also, what are the implications of this if the mice do develop this? So how the experiment works, it has several different parts. One of the first parts of it was to create mice who were knockouts for the gene that codes for nicastrin, to study behavior in these mice via different behavioral tests, which I'll talk about. Performing immunohistochemistry and electron microscopy on the mouse brains after they are dead, the mice who were knockouts for this, to look at their myelination patterns and see if hypomyelination has happened. And then to compare the results that they find from these mice to humans by looking for certain SNPs, genetic mutations in people with schizophrenia that are related to nicastrin, to see how do their nicastrin levels compare to the nicastrin levels of the mice and things like that. Okay, so here's some data from the behavioral portion of the experiment, which looked at the behavior of these mice who were knockouts for the gene that codes for nicastrin. So in one of the first experiments here, basically they took mice, the knockout mice and the wild type mice, and they put them through these different behavioral tests known as an elevated plus maze and an open field tests. So basically what those are is they put a mouse into a maze, or they put them into just an open area, and they observe their behavior and see what they do. So what they found was with the mice who were the nicastrin knockouts, they happened to sort of be hyperactive and ran around a lot more than the other mice, and they also had increased exploratory behavior, so they went farther and looked around. So basically they were less cautious than the wild type mice, which are behaviors that have been associated with schizophrenia. And then another experiment that they did was they tested the effects of haloperidol on these knockout mice, and haloperidol was first generation antipsychotic for schizophrenia, so it's not used as much anymore, but it was used as sort of a treatment of schizophrenia. And they wanted to see sort of what effect this would have on these mice, does it help, kind of testing more like if they're an accurate model of schizophrenia. And they found that it did change when these knockout mice were given the haloperidol. It did change their behavioral behavior to be more similar to the wild type mice. So they became a lot less hyperactive and they started to react more normally. So that's showing that this was somehow helping to restore the activity of nicastrin. And then finally, another observation that they made was that the mice who were knockouts for nicastrin, not only did they develop these behaviors that were associated with schizophrenia, but they also developed some behaviors that were associated with OCD, and they had some compulsive grooming issues to the point where they were actually pulling out their own fur and developing lesions from this obsessive grooming. So that's all of the data that they got from the behavioral tests. This all seems really overwhelming. So to summarize, nicastrin knockout mice display behaviors associated with schizophrenia as well as OCD. So that's kind of confirming the hypothesis. When they knocked out nicastrin, they did begin to display odd behaviors. And then they also looked at the hypomyelination in the mouse knockouts' brains post-mortem. So here's the results from that, and what they found out was that, interestingly enough, they only found hypomyelination in the optic nerve. So they were wondering, how might that compare to the hypomyelination patterns in humans who are lacking nicastrin, so like schizophrenic people, because it was believed to be basically more widespread in people than just the optic nerve. And what they found when they ran genetic testing for people with specific SNPs or variations of alleles that have to do with nicastrin in people with schizophrenia, they ended up finding out that in a lot of people with schizophrenia, they did have significantly altered nicastrin levels and lowered expression compared to people who did not have schizophrenia. And where did they find these lowered levels of nicastrin? They found lower than normal levels expressed in the blood and, most importantly, in the caudate, which is in a part of the brain that has a lot of dopaminergic pathways. So if you have hypomyelination in this area that is very important for dopaminergic projections, then that could mess a lot with the brain's dopamine levels. So that was pretty significant, and it's interesting that in the mouse model, that wasn't found. It was just found in the optic nerve. So there was a difference there. But overall, there was hypomyelination in both the mouse model and suggestions of hypomyelination in humans. I say suggestions because they couldn't actually test for the specific hypomyelination in humans, because obviously it has to be done post-mortem, and these people were alive, so all they could do was measure the levels of nicastrin, and then basically it implies that there's hypomyelination in those areas where it's expressed less at lower levels. So we don't actually know what we would have found if we could have gotten inside the people's brains, but that was not possible. So okay, so conclusions that we can take away from this experiment, there's two really important points that need to be made. One, hypomyelination as a result of the loss of nicastrin from gamma secretase may be a contributing factor to the development of schizophrenia. So why is this an important conclusion? It sort of paves the pathway of knowledge to discover more targeted treatments for schizophrenia. Because if you start to know that, schizophrenia, the dopamine imbalance is caused by this hypomyelination, you can start looking for drug treatments that will target this demyelination and start maybe look at like remyelinating, just like they would do with other disorders that have to do with demyelination and hypomyelination, like multiple sclerosis or things like that. So you could start looking at more targeted treatments and that would hopefully be more effective. And then the second important conclusion, hypomyelination as a result of the loss of nicastrin from gamma secretase may also play a role in the development of obsessive compulsive behaviors. So that's something that was interesting, because they were just looking for schizophrenic behaviors to begin with, and they ended up finding that these mice also had these obsessive compulsive behaviors with the obsessive grooming and the pulling out their so why is this important? It could possibly indicate a new subtype of schizophrenia. So what that means is that they could, if they find someone with schizophrenia who happens to also have compulsive behaviors, if they find that they have this lower than normal express levels of nicastrin, then they could say, oh, well, you need this specific targeted treatment that helps with remyelination. And so they would be treated differently than people who did not have these lower levels of expressed nicastrin. So that's important when it comes to treatment. And those are the major conclusions.